Good afternoon. I'm the Suwannee Riverkeeper, John S. Quarterman. That's a project and staff position with WWALS Watershed Coalition, Inc. That's a private 501c3 nonprofit. No, we're not associated with Florida State Agency Suwannee River Water Management District. We do talk to them all the time. We're sort of a thorn in their side. And WWALS has been around since June 2012. We've been Suwannee Riverkeeper since 2016. I've been doing that the whole time. I won't do it forever, but for now. And we do these WWALS webinars, usually on Thursdays, noon to 1 by Zoom. Today, we have a pair of distinguished professors from the Warnell School of Forestry and Natural Resources at the University of Georgia. They have some, I consider, exciting new research about an issue that people have speculated on for literally decades, which is how much water does the Okefenokee Swamp leak into the Floridan Aquifer? That's the aquifer from which we all drink and we depend on for industry, recreation, and pretty much everything in South Georgia and North Florida. Professor Jaivime Evaristo is an assistant professor of hydrology and water resources, and he works on the interaction of human and natural resources in water, air, and earth. And Professor Rasmussen, fluid flow and contaminant transport for surface and subsurface environments. They will introduce themselves further. And the two streams of research they have merged, which make for a very solid case. So I believe Dr. Evaristo will speak first. Over to you, Professor Evaristo.
Hi, good afternoon. Thanks, John, for the opportunity, and thank you all for your interest in the topic. My name is Jaivime Evaristo. As John said, I'm assistant professor here at UGA. So the short story is that Professor Rasmussen retired, and I was the replacement. And I came here straight from about 5 years in the Netherlands. And as any new professor in the area would have it, I was hungry for a project. And it turns out Professor Rasmussen has interest in the Okefenokee for quite some time. And my training is, I'm a hydrologist by training. And in terms of my tools, mostly I use tracers or isotopes. So sometimes people refer to themselves as isotope hydrologists, and I could probably count myself as one. And so it's just that I come with some unique set of tools and instruments, and there's this opportunity. And so this presentation or webinar is about essentially the paper that we publish about, or that's been published by an Institute of Physics journal in early December of this last December. So what you see here in the title slide is obviously the Okefenokee, and this was during our first field work back in February of 2025. So I'll start with how we present surface water, groundwater interactions in class by way of how we model them. So here is a cartoon, and it's supposed to be moving, but before I let the animation do the job, here you see a surface water body. In this case, it's labeled as a stream. It could well be a lake. And here is the land. You have some trees. There's a water table, and there's a pump right here. But notice that there is a layer here that's called the confining unit. And as the name suggests, it is a layer that's supposed to confine whatever water is below it from the water that's above it. So how we would present this in class and how we view and how things move in the subsurface with respect to this well that's installed right here is that water would start flowing from an area where there's higher head, so we call that the hydraulic gradient, so high to low. And as you can see, there's also some water from the surface water body here. It could be a stream or a lake, what have you, also being drawn into this straw, otherwise called a pump, that's taking water out of this shallow aquifer. Notice that there's a confining unit here, and there's nothing in the animation that suggests that there's any contribution or movement of water from the confining unit to that well right there. because if a confining unit is doing its job, which is to confine that water, then there should not be at least in principle interaction between this unit right here below it, the water below the confining unit, and anything that's above it. So here's a quick kind of animation of how we would present surface water, groundwater interactions in class where you have some human influence, in this case, a pumping well. So this talk is largely about the paper that I said earlier was published in Environmental Research Water Journal. And I would like to acknowledge my co-authors, Professor Jackson and Professor Rasmussen. Professor Jackson is still with UGA, and Professor Rasmussen just retired. If you're interested, you're more than welcome to Google the paper. There's also a QR code that's available in there. But briefly, where the study site is, I presume everyone knows where the study site is. But for those who didn't, this is the state of Georgia, and this is the Okefenokee right here in the blue outline. There you see several circles in there. These are different groundwater wells. The white circles are different groundwater wells that were sampled in 1997, well, 1995, around about that time, but published in a paper by Clark et al. In 1997 in Water Resources Research. So, and turns out there are 3 flow paths in this part of the state of Georgia. They call it, or we could refer to these flow paths as flow path 1, flow path 2, and flow path 3. In other words, this is where the water would go from the land into the coastal parts of southeastern Georgia. What was not done in that 1997 paper was they have not sampled anywhere near the Okefenokee Swamp, nor did they sample anything within the swamp itself or the swamp water itself. So our study did exactly just that. We used the data from the isotope data from 1997 paper of Clark et al. And we sampled a bunch of wells and surface water bodies and tap water around the area, around Okefenokee. And as you can see, there's also this flow path 3 right here. The graphs right here are the elevation maps that are shown in these flow paths. I would like to draw your attention to flow path 3 because this is the interest of this paper, the Okefenokee Swamp. So here is the swamp right here. As you can see, this tiny blip right here in this elevation cross-section profile. If you're familiar with the area, that's the trail ridge, right? So essentially, you can say that the trail ridge is kind of impounding or preventing that Okefenokee swamp to just flow to the other side. So that's the role of the trail ridge right there. So in terms of sampling, we sampled the swamp water, the Okefenokee Swamp water, some river water, and private wells that are open to the Upper Floridan Aquifer. In other words, they are municipal, and as the same is true for the municipal wells. Right. So this is the setup for the study. We have isotope data. I will talk about the tracer part of the study. Essentially, in the paper, we presented 2 lines of evidence. One is isotope-based evidence, and the other is hydrometric-based evidence, or water level fluctuations. And by water level, I mean water level time series data of the swamp and the Upper Floridan Aquifer. So that part of the hydrometrics or water level line of evidence will be presented by Professor Rasmussen in a while. So what's the status quo of hydrogeologic model of the Okefenokee and the Upper Floridan? So I showed this animation earlier, and there's this confining unit. The understanding is that, let's say this is the Okefenokee Swamp, is if you pump water past this confining unit, things should be fine. The swamp should be fine because there is that confining unit and it's supposed to be impermeable to anything that's above it, at least relatively impermeable. and that's nice, and that's true. That can be the case in a lot of places in the world. And the question really is that what would happen if your pump is way past that confining unit, as the case is if you were tapping into the Upper Floridan Aquifer, right? So I presented the preliminary results of this paper back in February of last year, I believe, at the Georgia Water Resources Conference here on campus at UGA. And I had a slide. It's exactly the same slide I'm showing right now, and I just summarized what's known about the area. Essentially, there are two quite influential papers about this part of Georgia. One was published in Science by Neil Plummer, who was with the USGS, and the paper I was talking about earlier by Jordan Clark and others in water resources research. So the consensus or the narrative, it seems, is that there is no connection between the Okefenokee Swamp and the Upper Floridan Aquifer. And I've asked people around, I've asked what other people think why this is the case, and they say that this is the case because the models show so. And what surprised me was that the models that were used to kind of characterize the surface, the hydrogeology of this region as part of Georgia are not based on any pumping tests or any slug tests, no data whatsoever. So I presented this at the Georgia Water Resources Conference, and somebody from the audience raised their hand and said, well, not yet in this slide, but in the slide I'm going to show earlier. So maybe I'll reserve that audience comment in that slide. So this is the status quo, or at least before a paper was published, of what the hydrogeology is of this part of Georgia. So as I said earlier, I'm going to present the isotope part. There are two lines of evidence that we presented in that paper. One is isotopes and the other is hydrometrics. So I'll do the isotope part. But what on earth are water-stable isotopes? Well, we could spend the whole semester talking about water-stable isotopes, but in the interest of time, the best description I could describe it, or at least make an approximation of it for general audience, is that they act as fingerprints of the different types or different sources of water. So what I'm showing you right now, so we can all agree that water is composed of two elements, hydrogen and oxygen. And there are isotopes in there. The isotopes of hydrogen and the isotopes of oxygen are presented in this plot right here. Oxygen on the x-axis, hydrogen on the y-axis. Now, why I'm using here tap water? I'm using tap water just an example because, like I said, stable isotopes of water act as fingerprints of different types or different sources of water. So here are different brands of tap water worldwide. You're probably familiar with Evian. And there's Dannon from Salt Lake City in Utah. There's Hawaii Volcanic from Hawaii. So depending on where the bottled water is sourced, that bottled water will reflect the isotopic signature of that source. Okay, so Hawaii waters being tropical would plot in this part of this kind of plot, a dual isotope plot. Most people call it or refer to this plot that way. And then if you're in a relatively snow and high altitude landscapes and more inland in Salt Lake City, Utah, it's more negative. Okay, this is much more negative, around minus 16.5 on the O18 relative to or compared to the -2 of the Hawaii volcanic water. Now, the reason why this is important, and why I thought that it's an effective visual for introducing the concept about water isotopes, is that imagine I take off the label of this Dannon Salt Lake City bottled water, and I paste it with a label of Evian, and I make you pay for the price of Evian, when in fact it's Dannon Salt Lake City water. We would probably pay for the price of Evian because the label says so. But once I run these waters in the lab, there's no way that you paid for well, there's a way that you pay for Evian, but there's no way that you're drinking Evian water because the isotope machine that's used to characterize the isotope signatures of different types of water would show that you are paying for the price of this water right here in Salt Lake City, Utah, for a probably a little bit more expensive price that you thought was from the Swiss Alps, somewhere in France, presumably. So this is the point here. The point of this slide is that isotopes of water can trace the different types and the different sources of water in the world and also in the landscape. And I'll show where I'll switch back to our study site by building on the same or showing the same figure or similar plot, a dual isotope plot with our data from the Okefenokee. So on to the data. So here is the similar plot that you saw earlier. This is the hydrogen isotopes on the y-axis, oxygen isotopes on the x-axis. Now, the bubbles or the circles here are the same color codes, or at least the same ones that are represented in flow paths 1 and 2 on the right side. So remember, these are from 1997, and they are in these parts of the study area, north of the swamp. Okay, so flow path 1 is north of the swamp, as you can see. Flow path 2 is also north of the swamp, as you can see right here. In terms of their fingerprints, where do they plot in this kind of presentation? They will plot right here in this region, in this cloud where my cursor is. I hope you can all see my cursor moving. And then the data from our study are the ones that are presented right here. We sampled twice, one in February, around Valentine's Day, and another in May. And they all plot right here in this area. Okay, and so these are the points, the locations of our sampling sites. So you can say that, so these are all groundwater Upper Floridan Aquifer samples. So right, let me clarify this a bit. These white circles are Upper Floridan Aquifer samples, as are these gray circles right here. They all tap the Upper Floridan Aquifer. The difference is that the white circles are from flow path 1 and 2, whilst the gray circles are from our study near and around the Okefenokee Swamp. This is the flow path 3. So, if you recall my tap water slide earlier, if they are all from the same source, why do we have two populations of waters right here? Right? If this is all Upper Floridan Aquifer, why is this more negative, a different fingerprint, flow paths 1 and 2, from these samples right here near and around the Okefenokee Swamp? So that's the puzzle. Because if they're coming from the same aquifer, i.e. the same source, recalling my slide earlier about the different bottled water around the world, then they should not result in two populations of samples, isotopically speaking. And if you recall the great movie of A Few Good Men, Tom Cruise asked this question, why the two orders, Colonel? So my question is here, why the two fingerprints of water if that is tapping from the same aquifer? Right. So how we could essentially, so we thought that there must be something interesting going on in this flow path right here that's different from these two flow paths. Well, visually, what you can see already is that flow paths 1 and 2 are quite far away from the swamp compared to flow paths 3. So something must be, and the landscape feature that's different in this picture right here is that flow paths 1 and 2 don't have the Okefenokee. The Okefenokee is way south of those two flow paths. So we did some mixing model analysis, essentially asking how much of those samples that of those water that we sampled in and around the Okefenokee Swamp is comprised of the Upper Floridan Aquifer. And here the sizes of the bubbles reflect the proportion of the UFA or Upper Floridan Aquifer water in those samples. As you can see, in general or in summary, the range is between 27 to 95% of the groundwater beneath the swamp is derived from the swamp. So I go back to my water resources conference experience. I presented the preliminary data of this study, and somebody in the audience raised their hand and said, I don't believe a thing of what you just said. I'm paraphrasing, but essentially it's quite a clear comment from a member of the audience, essentially saying, I don't believe what you're talking about. There is no connection between the swamp and the aquifer. And I thought, well, yeah, that's nice. At least I got some, interest from the audience. That doesn't happen all the time in a conference. So, but I just thought, well, at least we base our inference on some data from the site. We're not just we're not guessing or modeling based on how we view the world. So the key takeaway of this slide is that you have two different fingerprints of Upper Floridan Aquifer water. And the only difference between those two populations or two fingerprints is that one is farther away from the swamp and the other is closer underneath the swamp. Okay, so in this kind of tool, that is essentially how this works. You just would match it because, again, it's a fingerprint like you would in any fingerprinting. And we just look for the closest match between what you would expect as the fingerprint and what's the actual data or fingerprint data tell you. And the last slide on the isotope part before I give the floor to Professor Rasmussen is that we also could show some gradient with respect to distance from the coast. So here on the white circles are also data from the 1997 study. These are from flow paths 1 and 2. These are groundwater wells that are open to the Floridan away from the swamp. And you can see there's some sort of gradient of 0.6 per 100 kilometers to the coast. So you can imagine, you can just think of this as there's increasing hydraulic connectivity as you go from negative to more positive values of the O18 in the water. Now, our data from flow path 3 or from our study has a gradient of 1.1 per mil per 100 kilometers, right? So these white circles are here are flow paths 1 and 2 again, and these are from our study. So positive slope here or enrichment, as more technically referred to, towards the coast suggests hydraulic connectivity, but more so or almost double near the Okefenokee Swamp. Again, if you are tapping or drilling, or what do you call this, not drilling, if you are pumping water out of the same source, which is presumably the Upper Floridan Aquifer, the question is, why are there two signatures? Why are there two populations of isotopic fingerprints? So we suggest in that paper that is because of some influence or connectivity between the swamp, because that would be the most plausible explanation for the isotopic signature that we're seeing in the Floridan wells underneath and in the vicinity of the swamp. And we also presented our view of the hydrogeology and the high water balance of the swamp. So we estimate there's about 130 to 120 millimeters of leakage from the swamp to the Floridan based on a water balance approach. Now, and from a scientific perspective, there's uncertainty in each of these terms in the water balance. And the biggest source of uncertainty here is evapotranspiration, as is the case in a lot of places. So future studies would hope to constrain to understand evapotranspiration better by having measurements of evapotranspiration and not just leaving it as a missing term in your algebraic equation, which was the case here. So, but this is our view based on what we know now. Are there a lot of unanswered questions? Sure. Like any scientific research paper, there are a lot of areas for future research. But what I can just relate, at least, or convey to the audience is that here we present two lines of evidence. What I'm presenting now is the isotope part that characterize the hydrogeology of the Okefenokee Swamp. And I will give the floor to Professor Rasmussen, and I'll just leave you with this really nice quote, as I'm stealing it from my collaborator back in 2016, Professor Tina Roos from University of Southampton, the UK. She said that she wrote that model building must go hand in hand with experiments or data gathering. Otherwise, the models will just end up being sophisticated-looking Disney animations rather than scientifically rigorous and tested models. So I guess the takeaway is that models are fine and they can be very, very compelling visually. And they may look robust, but if they're not based on data, they're essentially just that, models. So with that, thank you for your attention. I'll leave the floor to Professor Rasmussen for the second line of evidence.
Hi, everybody. I'm switching over here if it will let me.
Professor Evaristo, oh, yes, you did. Never mind.
So I want to thank Jaivime. I came to Georgia in 1992 from Arizona. And Arizona used to have wetlands. It used to have rivers. It used to have lakes. But over a century of groundwater pumping, all of those disappeared. 98% of the surface water in Arizona disappeared due to groundwater pumping. The only 2% that are left are downstream of wastewater treatment plants. So when I came here, I thought, how could there be water left on the landscape? So 30 plus years ago, I made a beeline for Okefenokee because I wanted to see this feature of, just water everywhere. And it turned out that, I thought, well, someday people will want this water. Growth in Georgia and Florida is growing. And water is important. And so it'd be interesting to see what groundwater pumping might do to this. So I wrote a paper about the interaction between Okefenokee water levels and groundwater pumping. And that was in 1995. And it really didn't end up drawing much attention at the time, but for 30 years, I've always wanted to circle back and take a look at this system. So I think your WWALS area this is bigger than WWALS, is that right? This is from Sidney Bacchus's paper. I think this is the image on the left here is, if everybody can see that, is the Suwannee, and then the St. Mary's over here. And sort of in the center of all of this is the Okefenokee Swamp here. So the Okefenokee Swamp, when I came here, everybody said there should be springs that discharge into the swamp. But that's because they thought about all the springs in Florida that were common there. What I found is that the water level in the swamp under natural conditions is much lower than is much higher than what's in the aquifer underneath it. So in order to have springs coming up from an aquifer into the swamp, the Floridan here would have to be under high enough pressure to lift the water all the way up to the swamp. But as we'll see below, we'll show that really the water is flowing downwards. And so we represent this layer underneath the swamp that it essentially adjusts the flow. It regulates. It's like a valve that if you open the valve, you'll get more recharge. And if you close the valve, it reduces the recharge. So, but I painted here as this nice uniform color. My work at the Savannah River site, this is over near Aiken, South Carolina, between that and Augusta. It's not a uniform material. It's got all of these layers of clays, limestone, sands, muds. It's a complex media that is very difficult to encapsulate in a traditional hydrologic model. So what we do is we learned at Savannah River site is that we use this idea, we call it an anisotropy ratio. And all that means is that the permeability in the horizontal direction, or how quickly water flows horizontally, is different than the rate at which it flows vertically. So I think of it like an interstate highway. If you're driving down the freeway, you can get pretty good speeds. But if you try and cross perpendicular to the highway, on the highway, you don't have a bridge or a tunnel underneath it, but you got to get from one side to the other. It's much harder to flow in the opposite direction. So what we say is that the horizontal ability of groundwater to flow is 1,000 times what it is that it takes to flow vertically. And so the Okefenokee Swamp is up here in this lower corner, is much higher than the Floridan Aquifer. And so what happens is water is trying to flow downward at the same time it's trying to flow horizontally. And so what it says is that water that leaks from the swamp, some of it's going to end up in the aquifer under the swamp, and a lot of it's going to flow over here to the St. Mary's River, essentially just simply due to the fact that it's more permeable in that direction. There we go. So the only way to characterize this is to get data. So we have a couple of wells here, and at Stephen Foster State Park, the U.S. Geological Survey has a well there. There's another one up here near Waycross, one near Brunswick, and a bunch near St. Mary's here. So what I've done is I collected, this is on the internet, you can download this data. And these are, if you were to take a mole's eye view, if you were to look at it from the side, this is away from the ocean, and this is down on the coast. And this 27E004 is the well that's directly under the swamp. It goes down about 700-800 feet. There's another one up near Waycross. It goes much deeper. It goes almost 2,000 feet down. The wells over on the coast, they're at different depths. So for example, 74 goes way deep, 73 doesn't go as deep, 71 and 72 are up here near the surface. And this 33 is up near Brunswick. So what this does is it gives you small little windows into the subsurface, and you can take a peek at what is going on there. So if you look at the data from about 1980 in each of these wells, this top one here is the Upper Floridan Aquifer underneath Okefenokee Swamp. And you can see the groundwater levels are at about 50 feet above sea level. Okefenokee Swamp is higher than that. It's about 30 feet higher, up to 60 feet higher. And so this water level in the Upper Floridan is, the Upper Floridan here is the purple line. And there's another one here in the lower Floridan, which tends to track the same. So what you're seeing is that the water levels are not constant in time. Now, if you go down to the coast, you'll see this weird behavior where water levels were way deep, and then all of a sudden in 2002, they came up and settled into a new normal. And that's when the Gilman Paper Company shut down. They used to have a pulp and paper plant down there at St. Mary's that shut down in 2002. And when they stopped pumping, all the water levels in those coastal wells rebounded. Now, what is this? 10, 5 to 35, 30 feet. This Floridan aquifer well responded. And you can see there's continues to be noise here, but there's an important response that you're seeing there. And so what we do to look at this is, let me go put my surface water hat on. Essentially, when we get rainfall, we see streams rise and then fall down. We call this a hydrograph. And a hydrograph is essentially just telling how streams respond to precipitation. And you can see that this rainfall in the solid takes some time for there to be a response in the stream. If it rains today in Minneapolis, you don't see the river in New Orleans respond immediately. It takes a long time for the water to flow from Minneapolis down to New Orleans. So what we're doing is something similar. We look at how long it takes for the water levels in the aquifer to respond to a change in pumping. For example, when they shut down the wells in the lower Floridan, we'd like to see how long it takes for the upper superficial aquifer, the one right next to the surface of the ground, to respond. And so what happened is, when they shut off the well in St. Mary's, the groundwater in the lower unit changed quickly, but it took about 50, 60 days for it to fully respond in the shallow aquifer, meaning that you change pumping down below underneath the swamp, and it makes an impact, say on average about 30 days later. So what we did is the same thing now. What we're trying to do is say, okay, how long does it take for some change in the swamp to affect the Floridan aquifer underneath the swamp? And essentially, it's just like the other one, but in this case, rather than the effect being from the deep aquifer up to the shallow, we're looking at the effect of the swamp on the Floridan, because this is the one that's recharging the Floridan. And you can see that 114 is the sort of the hydrologic, the water level in the swamp, and 45. So that's a difference about 50, 60, if I do my math, 70-foot drop. I mean, that's a huge drop. The water level drops 70 feet from the swamp down to the aquifer. And so this lower water level is pulling water from above. And we'd like to see how long it takes for the water level in the aquifer to respond to a change in the swamp. And so when we do that, we say, okay, if the groundwater, or if the water level in the swamp changes by a foot, how long does it take for the water level in the aquifer to respond underneath it? And again, we see something very similar to what we saw down on the coast when the pumping stopped at St. Mary's. Essentially, after 30 to 60 days, you'll see that the water levels in the aquifer change due to changing water conditions on the swamp. It'd be like if you had rainfall at a certain place. How long does it take for that stream flow to respond? In this case, we're saying, what does the water level in the swamp do to the water level in the aquifer? And because the water is flowing downward, there's going to be a delay from the swamp onto the aquifer. So all I'm saying is that from the hydrologic point of view, hydrometric is what he's saying, is that the idea is that if you see a change in water levels at one point, how long does it take to affect it at the other point? So the U.S. Geological Survey was told by the state to go conduct aquifer tests along our Georgia coast. And they went out and they said, okay, we're going to pump the aquifer for 2 days and see if there's going to be effect on the surface water within 2 days. It was a 48-hour test. And my argument is that you're never going to see a response in 48 hours. It's going to take months for you to see a test. And so the whole notion of how long or what is the time constant, what is the time delay between pumping and their effect on groundwater is much slower than what most folks have in mind. And so the notion that the gradient is downward, that it affects St. Mary's River flows, and that these are all showing a response due to stable isotopes. And I think I'm 1 minute over, but let me just say one more thing. The stable isotope concept comes from the idea that hydrogen has two forms. It's normally simply proton, but occasionally it has a neutron attached to it. And that's what makes heavy hydrogen, or heavy water, as you might say. And if you take a pot of water and put it on the stove and you boil it, all the lighter forms that just have one proton boil off faster, and the heavier forms take more energy to boil off, so they boil off slower. So over time, it gets heavier and heavier on the stove. That's why something like the Hawaiian drinking water is so much heavier than the ice form, is that it's just been evaporated down so much. And if there's one thing we can say about Okefenokee Swamp, is there's a heck of a lot of evaporation going on there. And all that evaporation is making both the hydrogen and the oxygen heavier and heavier. It makes a very clear imprint or fingerprint on the groundwater underneath it. And I should say also that oxygen has two forms. One is normal oxygen has 8 protons and 8 neutrons. Occasionally it has 2 extra neutrons. So that would be heavy oxygen. So the plots that Jaivime was showing showed both a heavy hydrogen enrichment and the heavy oxygen enrichment. And so you would expect that from anything where you undergo severe evaporation, which is consistent with what we see in the swamp. I'll stop there.
All righty. Thank you, professors. We've got one question here in the chat, which is concerns or issues with the quality of the water leaking from the Okefenokee Swamp into the aquifer.
Do you mind if I put? So the idea is the rate at which the water flows is much slower than the pressure response. So when the water level goes up in the swamp, it's a pressure response. I explain that by saying, well, let's say you have a hot water heater and you have a pipe from your heater to your shower. When you turn on the shower, you get a very rapid pressure response. But the hot water takes much longer to go through. So in our case, we have a very long pipe. It's going to take who knows how long, hundreds of years maybe, for the water from the swamp to filter through those sediments. The hydraulic response, like when you turn on your shower and it pops out instantly is a much faster response than the actual water movement. So whatever effects in the swamp are going to be mitigated by the long travel times that it takes for the physical water to move through the aquifer.
So if there's contamination, it probably hasn't actually gotten to the aquifer yet because it takes so long.
Exactly. So, for example, when you look at a stream and you see the rain flowing in the stream, in a city, how long does it take for the water to run down the street and flow into the stream? That's pretty quick. If you look at how long it takes for the water to flow through the subsurface, say you have a forested watershed, and it soaks into the soil, it recharges groundwater, it may can't come out 5, 10, 20 years later, or even 20,000 years later. It's just much slower for water flow. The hydraulic response, it'd be in groundwater, could be very rapid. It could be on the matter of hours or days, meaning it'd be like turning on your tap in your shower. That hydraulic, the pressure pushing through is so much faster than the actual water flowing through the subsurface.
Which, of course, could also mean that if there's contamination going through, it's going to show up quite a bit later and be a big problem and be very hard to fix.
Exactly. Back in the 1950s, they said that the earth was a magic filter, that you could dump any waste on the ground that you want and it would get totally filtered out. Well, they just didn't wait long enough. All these Superfund sites showed the problem with that.
I have a question. It's vaguely related to when the paper mill shut down. You've probably heard that Chemours is closing and has closed several of its titanium dioxide mines. For example, the one they had near Jessup actually closed in November, and they have closed at least one of the mines in North Florida, not all that far from the swamp. I don't know how much water they use, but probably a significant amount. I wonder if you can already see any effect in the water pressure or levels underground.
Well, that's a great question. I haven't looked at recent data. I keep an eye on that. To me, that's a good question I see these wells as sort of thermometers that measure the pulse of the aquifer, right? And we have so many federal agencies that collect this data, but very few people who actually look at the data and interpret what it's going on. Like, how many people keep an eye on their speedometer these days? I swear nobody ever looks at their speedometer.
Yeah, good point. We have another question here. Just one moment. Oh, this is from a reporter from WABE. This is for Dr. Evaristo. Would you be able to pronounce your name just so we have it correct for radio?
Well, yeah, thanks you, Marisa. My first name is Jaivime, like Jaime, the Spanish J pronounced as H. So Jaivime Evaristo.
Okay, apologies for getting that wrong earlier.
No worries.
And her substantive question is, could you speak to the methods of this study and how it compares to previous research like that relied on by the EPD and Twin Pines that asserts the swamp is not connected to the aquifer? For the layperson, how does this study differ from previous studies?
Yeah, I'll let Professor Rasmussen to speak more to that, but I can answer the first half of the question, how it compares to the previous studies. So there are just a few studies done in the area, nothing around near the swamp. So like I showed earlier a slide, there are 3 flow paths. The 1997 study using isotopes, also like the same tools that we use for our study, show that those aquifer Floridan aquifer water are isotopically different from the ones from the Floridan aquifer water underneath the swamp or within the vicinity of the swamp. And I always tell my students when I talk about isotopes, like if you have a container, a bathtub, or even a small, let's say, water jug, and you have two straws, you would expect the identity, the fingerprints of the water that comes out of those two straws to be the same because they're coming from the same water jug. What we're finding in this system is that the same water jug called the Upper Floridan Aquifer has two different fingerprints or two identities. Someone has got to explain why that is. And if you look at the landscape feature, the only difference between those two populations is that the presence of the swamp. So isotopically speaking, there is that connection between the swamp and the Floridan aquifer. From what I know, based on the EPD and Twin Pines, like I said, Professor Rasmussen would know more about those things, is that in general, regardless or beyond those EPD and Twin Pines report or models, etc., none of those that I know of is based on data from the site. So to the extent that you want to bet one's farm on models, I guess there's that question as to perhaps we could just go out there and collect some samples. Perhaps we could just look at water level fluctuations like Professor Rasmussen just talked about a while ago. And it's not that much, I can tell you. It's very cheap to analyze water for isotopes. You just have to go out there, do some work of pumping some wells and collecting some data, and should be at least you have data-based support for informing future models. So I'll give the floor to Professor Rasmussen. I'll stop there.
Yeah, I mean, the whole notion of, doing additional efforts, the state never made Twin Pines look at the influence of the connection. That was just assumed throughout, that there was no connectivity whatsoever. And it was based upon U.S. Geological Survey studies based on 48-hour pumping tests that showed that there was no response. And the argument being that 48 hours is not enough time to demonstrate a response. You need to go much longer than that. And this has implications throughout the southeastern coastal plain. I hear these same statements that the Floridan has no connectivity to surface water in these areas. Up at the new facility up on I-16 outside of Savannah, even, they're making that claim there, even though the facility is right next to Sinkhole Road, and there are sinkholes everywhere in that system. And the whole notion that they're completely disconnected defies scientific understanding. Without any kind of data to support it, meaningful data to support it, it's just the evidence should be thrown back on the applicants to demonstrate they have the data to show that this is not true.
They could have just looked across the state line because the Swanee River Water Management District and the St. Johns River Water Management District have extensive data and models based on them showing how pumping affects various levels of the aquifer. And in case anybody doesn't know, by far the biggest pumper and therefore the biggest effect comes from the city of Jacksonville. In the Swanee Basin, most of the withdrawals come from agriculture, but there's more than all of agriculture in the Swanee River Basin pumped by the city of Jacksonville, which is not that far from the Okefenokee Swamp.
If I could say that Dr. Jaivime's analysis could tell you what percentage of the water they pump in Jacksonville comes from the swamp.
Oh, good point. How can we get a study on that done?
I think we just have to go down and grab a tap water sample from Jacksonville.
I'd be for it. I'd even bring it to Professor Evaristo if that's what it takes.
Yeah,
Well,
happy
he's
to
very
help.
particular in the way he samples. Waycross, I don't know what other urban areas around there would be interesting to compare. And, just look at this isotopic signature and just tell what proportion is consistent with the source in the swamp.
Right. Well, and Valdosta as well. There's actually a USGS uses Valdosta as a case study in one of its publications. There's a sinkhole west of the Withlacoochee River. The river water goes into it, goes under the river and several miles to the east to where Valdosta's water wells are. And they had a choice of either spending a lot more to deal with the biological things in the water and the acids, botanic acids, or just sink the wells twice as deep to get under it. And that's what they did. So, even in Georgia, it's well known that pumping and surface water interact all the time. It just astonished me that EPD did not deal with that. We have few more questions here. Should we now no longer allow any new water permits, Florida and or Georgia, from any new developments, pull water from the aquifer and or from adjacent rivers? I think you're asking a policy question of a couple of professors, but we'll see if they'll answer.
What I'd like to say is that in Southwest Georgia, they've developed this great program down there. It's called Georgia Fit, Flow Incentive Trust to manage groundwater allocation. The state had developed different zones, red, yellow, and green, in order to prioritize evaluating permits. And to me, it would be nice to develop a policy that's already in place, or to emulate a policy that's already in place and apply it to coastal Georgia. It would be, if they have something that works there, it would be nice to consider something of similar nature along coastal Georgia, or coastal Florida for that matter.
Wasn't there a study some years back when there was an issue with overpumping on the coast of Georgia, and the state actually funded a study to study, I can't remember the name right now.
The Sound Science Initiative?
Yes,
yeah, that was the one where they required a 48-hour aquifer test.
Right, which wasn't adequate, but at least it's an example of the state can require this and even fund it. It would be better than waiting on, for example, mining companies to fund this sort of thing, which, anyway. We have another question here. Is the trail ridge just a surface watershed feature for the swamp, or is it also critical for flow path 3 to recharge the Floridan Aquifer?
No, on my first slide, I showed streamlines, which showed how the water flows through and under trail ridge. And to me, it would clearly, our data are not at the high enough resolution to look at specific flow paths. I was hoping a study by like Twin Pines could do something like that. Since they're going in and developing the site, they had all these wells. My recommendation always was that it'd be nice to do that kind of analysis. But, the state was certainly not willing to require them to do that.
Very unfortunate. Another, if I may make a historical analogy, the states seem to be depending on Ptolemaic models of water flow, like Ptolemy had epicycles to explain the movement of the planets, Whereas you guys actually collected real evidence and built a model based on that. So I don't know which of you is Copernicus, but I thank you for doing it. Another thing they just assumed was, you're probably aware that the swamp is said to have basically 4 major basins. And they just assumed that, contaminants would not travel amongst those basins, which is like, excuse me, you can see the water flowing. Also, 85% of the swamp actually drains into the Suwannee River. So as you can imagine, as Suwannee Riverkeeper, we're quite concerned about all this, plus the aquifer underneath from which we all drink. Do we have more questions for the professors?
Well, could I respond to that?
Oh, yes,
What's
please.
interesting is that the refuge actually has water level data and rainfall data going back to 1941. And they have a treasure trove of dozens of monitoring sites throughout the swamp that they've collected. I would call a treasure trove of information that is just waiting for someone to look at, at some point I'll probably get around to sort of digging into this, but it's like an overwhelming data set that can answer many of these questions that nobody has ever looked at.
I'd be happy to look at it, but I do not have the resources of a university professor nor the knowledge. So if there were one who wanted to look at it, that would be great. Professor Evaristo, it looked like you wanted to say something.
No, completely agree. As esoteric as isotopes might sound, they really are not, but they're very powerful. It used to be very expensive to run them, but now they're very cheap. So cheap that I even ask my students to collect tap water from all around Athens and analyze them for the samples for them, and then we talk about it in class. And they can be very powerful. So if you have any interest in tracing water or quote unquote fingerprinting water of all types, not just not just swamp water, let me know, and I'm more than happy to. Don't think about the cost. Just let me know, and more than happy to cover the cost for you.
Oh, well, we have interest in knowing things like how much water in the Suwannee River is coming from upstream and how much is coming out of that outflow pipe from effluent from Pilgrim's Pride, and how much is coming from the city of Valdosta, how much is coming from cattle farms, things of that nature.
That's perfectly in my wheelhouse. That's
Yeah,
great.
a lot cheaper than DNA tests, I bet.
Oh, yes. Yeah.
So
Sure.
interestingly, I found a report by the State Geological Survey in the 1950s where they went around the state and they used water quality composition like calcium, sodium, sulfur, all those things to identify the sources of water in all the major rivers in the state. And I'm going, what a wonderful study, but it's never been repeated.
That's kind of like the state tests every river in the state twice a year, whether it needs it or not, which is totally useless for our purposes of trying to find out where the contamination is coming from. We have a water quality testing program. We mostly test for E. coli, and we've managed to find several sewer spills and other sources. Turns out cattle in Brooks County, Georgia were a big problem. They have improved their act. Looks, Nancy Loeffler, are you trying to say something? If so, you're muted. I guess she's not. Okay, well, we're approaching 10 minutes after the hour. Has anybody got any last minute questions, or we should let the professors have some lunch?
Or do they know how to get a hold of us? If they want to email us somehow?
I believe your contact information was in the Facebook event. If not, I will add it. You can also just put it in the chat. And of course, your email addresses are on your UGA pages.
Well, they got rid of me as soon as I left.
Okay. You still have a UGA address though, don't you?
Yeah.
Which is something like Todd.Rasmussen at,
Rasmus.
yeah. We do have one more question. Will the presentation be sent out after this?
I can put a link to my, the recorded.
The recording, the Zoom recording will be available on YouTube. It may take a couple of days. We're a tiny little nonprofit, so it takes a little while. Professor Rasmussen has sent me his presentation, and he's offering, I think, to make a link to it on his website. Professor Evaristo, do you mind if we share your presentation? All right, if you want to send the slides, we will do the same with those. And they have both put their email addresses in the chat, trasmus@uga.edu and jevaristo@uga.edu. And just to wrap up, here's a comment. Thank you. This was an excellent presentation. Even this English major could understand it. I thank you both for the research, for the presentation, and sounds like you're going to do even more research, and you're enabling others by pointing out all these sources of data and methods that turn out to be much less expensive than expected, like isotopes.
Thank you for the opportunity, John. Thank you all.
Thanks everyone for attending, and thank you, professors.